Welcome to the Academic Express. We begin with a newly laid, warm egg. Floating on the surface of the yolk is a single, translucent disc of cells called the blastodisc, which contains the genetic instructions for the entire organism. Development initiates with syngamy. The male and female pronuclei drift toward each other within the cell. Their membranes dissolve, and their genetic material merges into a single zygote. Immediately, the zygote begins to divide. Cleavage furrows slice through the cytoplasm with geometric precision, doubling the cell count from 2 to 4, then 8. This process is managed by microtubule spindles. These structures physically anchor and pull the chromosomes apart, ensuring each new cell receives an identical copy of the genome. By the end of the first day, the blastodisc has become a mound of identical cells called blastomeres. This cluster provides the raw biological material required for the specialized tissue organization that begins in the next phase. On day 2, the cells begin to organize spatially. They sort into a transparent central zone called the area pellucida, the site of the future embryo, and a darker outer ring known as the area opaca, which will form the support tissues. Chemical gradients of nodal and VG1 define the cranial-caudal axis. On day 3, the primitive streak appears, directing gastrulation. In cross-section, epiblast cells undergo an epithelial to mesenchymal transition, sliding through the primitive groove to form mesoderm and endoderm, resulting in a 3-layer embryo. Henson's node sits at the front of the streak, secreting proteins like chordin and noggin. These chemicals block local bone morphogenetic proteins, which allows the nearby tissue to begin forming the brain and spinal cord. The formation of this trilaminar disc establishes the body's primary architecture. The embryo's orientation and basic tissue types are now physically set. As the primitive streak recedes on day 4, it leaves behind the notochord. This is a stiff, liquid-filled column of cells that provides a temporary structural scaffold for the developing body. The notochord also serves as a signaling center. It releases a protein called sonic hedgehog, which diffuses toward the overlying ectoderm to instruct it to differentiate into ventral nerve cells. Simultaneously, blood islands appear in the area opaca. Here, specialized cells called hemangioblasts begin the assembly of the first embryonic blood vessels and red blood cells. These chemical signals and physical structures establish the internal framework necessary for the embryo to begin folding into a 3-dimensional form. On day 5, the flat disc begins to fold. The head lifts, the lateral sides curl inward, and the embryo assumes a distinct C shape. Inside the body, a simple heart tube forms and bends into an S shape. Calcium waves travel through the tissue, triggering the first rhythmic contractions of the muscle cells. Along the spine, blocks of tissue called somites appear. These segments are the precursors to the vertebrae, the skeletal muscles, and the deep layers of the skin. On day 6, the neural tube swells into 3 primary vesicles, governed by a specific Hox genetic code. Zooming in, stem cells divide rapidly. Their nuclei move up and down in interkinetic nuclear migration, driving high-density neuron production. On either side of the head, the optic vesicles bulge outward to begin the formation of the retinas. The initiation of the heartbeat and the rapid expansion of neural stem cells transition the embryo into a stage defined by active cardiovascular and neurological systems. By day 7, wing and leg buds emerge. These are driven by specialized signaling at the tip of each bud, which keeps the underlying cells dividing to extend the limb. Neural crust cells migrate from the spinal area into the pharyngeal arches of the neck. These cells provide the structural material for the bones and muscles of the face and throat. On day 8, the embryo is fully enclosed by the fluid-filled amnion. Internally, the liver bud grows and the primitive kidneys begin to form along the trunk. The lungs begin as 2 small pouches branching off the foregut. They expand into the surrounding tissue in response to local growth factors. On day 9, specific signaling loops trigger the formation of feather placodes on the skin. Simultaneously, programmed cell death, or apoptosis, removes tissue between the digits to sculpt the individual toes and wings. In the abdomen, the gonadal ridge forms. At this stage, it remains indifferent, waiting for the hormonal signals that will determine its development as either ovaries or testes. From the lungs to the feathers, these anatomical features are sculpted through localized chemical signaling and the precise removal of excess cells. By day 10, the beak develops a temporary hardened egg tooth. The claws also begin to solidify through the production of beta-keratin. Ossification begins to take place within the developing skeleton. Osteoblasts deposit bone minerals over cartilage templates, while osteoclasts hollow out the centers to create marrow cavities. This diagram shows the allantois, a vascular membrane that sits against the inner shell. It functions as the embryo's lung, exchanging oxygen and carbon dioxide through the microscopic pores in the eggshell. On day 11, the retina matures into distinct layers of light-sensitive cells and neurons. Even in the darkness of the egg, spontaneous electrical pulses test the wiring of the visual system. The eyelids fuse shut for protection, and the first pin feathers emerge, while leg scales overlap like tiles. By day 12, the embryo has established its primary life support interfaces and sensory foundations, preparing it for the transition to the outside world. On day 13, feather barbs and barbules branch out inside their protective sheaths. This branching pattern creates the complex structure of the down feathers. The membrane surrounding the embryo begins to dissolve small amounts of calcium from the shell. This mineral is transported into the bloodstream to harden the growing skeleton. By day 14, unique avian features like the pecten are fully functional. In the brain, the neural centers for future vocalization begin to connect. The embryo is now highly active, rotating within the egg to exercise its muscles, preparing for the demanding physical effort of the impending hatch. The immune system also matures as white blood cells begin to populate the spleen. On day 15, soft down feathers cover the entire body. The chick begins to practice breathing movements, contracting its chest muscles against the fluid to build strength. With its organs and muscles developed and tested, the organism has the energy reserves necessary for the physical effort of hatching. On day 16, the chick moves into the hatching position. It tucks its head under its wing, with the beak and egg tooth aimed toward the air cell at the blunt end of the egg. During day 17, the remaining yolk is drawn into the abdominal cavity through the yolk stalk, providing a final concentrated source of energy. This yolk transfer also provides the chick with maternal antibodies, granting it passive immunity to protect it during its first days of life. The lungs are now fully formed. They utilize a cross-current exchange system, where air and blood flow in directions that maximize the efficiency of oxygen absorption. This final stage of nutrient absorption and respiratory maturation prepares the chick for immediate biological independence upon leaving the shell. On day 18, internal pipping occurs. The chick's beak pierces the inner membrane, allowing it to breathe the air trapped inside the air cell for the first time. This intake of air triggers a cardiovascular shift. A sudden rise in oxygen causes the ductus arteriosus to constrict and close. This shuts down the fetal bypass, rerouting all blood flow through the lungs, establishing an adult circulatory pattern. As the chick begins to chirp inside the shell, the external blood vessels in the allantois begin to dry up, as they are no longer needed for gas exchange. A single intake of air has initiated a permanent physiological change, transitioning the chick from a membrane-reliant embryo to a lung-breathing organism. External pipping begins on day 19. A specialized muscle on the back of the neck drives the egg tooth to violently crack the outer shell. On day 20, the chick rotates and chips a circular line around the shell. This zipping process is stimulated by the rising levels of oxygen and carbon dioxide in the air cell. On day 21, a final physical heave pushes the shell cap away, and the chick tumbles into the open air. As it dries, the chick's down feathers fluff out to trap heat. The lungs fully inflate, and the remaining yolk inside the abdomen provides nourishment for its first hours of life. These 21 days track the transformation of a single cell into a sentient, breathing creature. This progression, from the first chemical gradients to the physical effort of hatching, concludes the transition from a genetic blueprint into an independent organism.